see when you put your hand up.
In a polite and courteous way, of course. Yeah. Right.
Yes. Okay,
Mary,
thanks, Steve.
I'll hand over to you. You're being recorded. So if everyone mute, please, and then full screen, and then away we go.
Okay, thanks, Steve. So this lecture continues some of what we've already talked about in this module with regard to glycopeptide antibiotics. You will have heard a couple of 2 or 3 lectures, either from Steve or me, relating to glycopeptides. So this lecture deals more with the industrial side of glycopeptide antibiotics and their production on a sort of commercial level. So this front slide is just to illustrate a little bit of PR that was put out by Novartis, of whom you may have heard, a pharmaceutical company who are just expanding their production and manufacture of glycopeptide antibiotics, amongst other antibiotics, in Austria. It's one of the largest facilities for manufacture. And this coming at a time when antibiotics are sorely needed as accessory treatments during a pandemic. Okay, so we're talking about glycopeptides in keeping with this module on glycoproteins and polysaccharides. So vancomycin and teicoplanin are clinically used natural, i.e. first generation, glycopeptide antibiotics, much used. And there is increasing use of some second generation derivatives of those that are semi-synthetic, oritavancin, telavancin and dalbavancin, that are lipoglycopeptides. They have these lipid-like chains attached to them, like teicoplanin really. Teicoplanin also has that. And when these antibiotics, these lipoglycopeptides act, they insert themselves into the bacterial membrane. But otherwise, they act as the parental vancomycin does in the Pepto-Glycan of bacterial target cells. And I'll come on to that in more detail in a moment. Okay, so this is what I'm intending to talk about. This is my plan. So I was thinking more broadly initially about antibiotic targets in bacteria. Various antibiotic classes exist, of course, and some of you will be familiar with the various classes of antibiotics and what they target in bacteria and why they're good for targeting bacteria without harming the host. If you haven't had that background, then I've put a few slides in an appendix at the end of this PowerPoint presentation so that you can have a look, if you're interested, more broadly at antibiotic classes. I'm going to talk briefly about antibiotic consumption in the UK in particular, including the glycopeptides, of course, the mode of action, and then come on to the industrial process in how it's produced. So the producer microorganisms, the concept of secondary metabolites, scale up, what do we mean by large scale, and then how antibiotics are then purified out of those fermenters. And then I'm going to talk about vancomycin specifically because we've only got one lecture. I could go through all 5 of them, but that would take far too long. So I'm going to mostly concentrate on vancomycin to talk about not just the fermentation process, but how people have tried to improve it. So optimizing wild type strains to get better yields, and then strategies for improving classical strains that are still used today, And then other strategies like ribosome engineering and genetic engineering strategies for producer strains of glycopeptides. So that's the plan. So to begin with what we consume by way of antibiotics in the UK, if you were expecting me to say, well, antibiotics are required more and more and more, that's partly true. But in terms of the overall trajectory, there is a downward trajectory in terms of consumption of antibiotics. And the reason for this, you will quickly see from the last, that video lecture that you've just looked at, is because one of the strategies for overcoming the antimicrobial resistance crisis is for us to stop using antibiotics unless they are absolutely needed. So GPs, veterinary surgeons have all taken action to cut their usage of antibiotics. Yes, if they're needed, they're prescribed, but useless, senseless prescription of antibiotics that aren't going to cure, for example, virological diseases has been cut. And you see that in the stats. So the trajectory is downward. But if you break it up into veterinary usage of antibiotics compared to clinical use in humans, you see that actually there is an increased usage in people by 8%. And it's the decrease in veterinary circles, a cut of 51% in agriculture, which is contributing to this overall downward trajectory. Okay. Here, of course, down this table of different antibiotics are our glycopeptides, and you can see that consumption is confined to human use and none in agriculture. In 1997, it was suspected that usage of glycopeptide antibiotics to boost animal production, not only to cure diseases, but to increase the mass of animals, and was having an effect in terms of we were seeing an increase in glycopeptide resistant bacterial strains in the clinic. And the link was made to the fact that glycopeptides are used in agriculture. And these glycopeptides are generating glycopeptide resistant bacteria, which find their way through the food chain to humans and to the clinic, and were responsible for generating glycopeptide resistance in the clinic. So in 1997, it was banned in the EU, including the UK, of course. And it's a human-only antibiotic now. There is some controversy as to whether that transfer of resistance from animal usage to the clinic was actually real or not. And in the literature, you'll find controversial, conflicting evidence that it was actually so. But nonetheless, the ban is in place, and glycopeptides are used specifically to treat human infections, not animal infections. The stars down the side of this table just depict the antibiotics that have been singled out as the highest priority antibiotics that are used in people, though for some of them it's recognised that you have to continue to have their usage in animal circles, but they are singled out as priority antibiotics for treating human infections, and glycopeptides comes into those categories. And in fact, 89% of these highest priority antibiotics are used in people. So that segregation into what you can use in veterinary circles compared to human circles in the clinic is really taking effect. And even within a class of antibiotics, there are some members of that class which are specifically used for animals, and then other members of the same class that are used specifically only for humans. And that's not to do with generation of resistance or anything. That's to do with marketing and availability to the two different sectors, I think. Okay, right. Let's look at more at vancomycin is the predominant glycopeptide used in the clinic and its consumption. And if we break down when it's used in two different forms: an injectable form to treat MRSA, methicillin-resistant Staph aureus, and other penicillin-resistant infections. So it's injectable or it's infused. So this is shown for major EU markets and the US altogether, that by far the greatest amount is used in the injectable form. But there is also usage in an oral form, which is to treat pseudomembranous enterocolitis. So this is an infection in the gut. It's attributable to Clostridium difficile, a gram-positive anaerobe in the gut, which tends to predominate when patients have been kept on antibiotics long-term. So antibiotic action not only kills infections, it kills commensal organisms in the gut as well. And this allows Clostridium difficile to predominate and cause a problem. This is a predominance of C. diff causes diarrhea and this invasive enterocolitis if not taken in hand. And vancomycin, when taken orally, does not cross the gut wall into the bloodstream. It remains in the gut and it finds its way to the intestines where Clostridium difficile predominates, and vancomycin is effective against gram-positives predominantly because it inhibits in the Pepto-Glycan and cannot cross the outer membrane of gram-negative cell walls. So it's very good against gram-positives, including MRSA, as we've mentioned. That's gram-positive as well. Okay, in the UK, I've compiled the analogous consumption data. It's a bit old now, but it does show you that we are still on an upward trajectory in terms of glycopeptide use. And this is largely because we've increased our infused glycopeptide usage through teichoplannin by 25% quite recently in England. So we're on an upward trajectory in terms of using glycopeptides in the UK. Whoops. Okay, I deleted one of the slides, so you might be confused when you look at what I've uploaded onto Moodle. I just deleted one slide because we've already had that slide. Anyway, so just to fill you in, the mode of action of vancomycin is, I've already mentioned that it inhibits, the target is in the Pepto-Glycan layer of susceptible cells. And we have moderate interactions of vancomycin with its target. And the target is shown here on the left-hand side. It is the diala depsipeptide of the lipid 2 unit of Pepto-Glycan. So if you remember back to your undergraduate days, I'm sure a lot of you will have learnt about the structure of Pepto-Glycan. That we have a lipid carrier back to prenol. It's a C55 alcohol which lives in the membrane and takes Pepto-Glycan units, which is basically this part of the molecule, from the inside of the cell where it's manufactured, flips it over, it's called a flipase, across the membrane to the outside of the cell wall, outside of the membrane, and it's inserted into the peptidoglycan layer on the outside of the membrane. So this is a unit of peptidoglycan, if you will. And it comprises N-acetylglucosamine linked by a beta-1,4 link to N-acetylmuramic acid. And the muramic acids have pentapeptides attached to them, which become tetrapeptides during cross-linking with neighbouring glycan strands. So here's our glycan strand. There is a process called transpeptidation, whereby this terminal dialer is cleaved off, and this allows a peptide bond to form between this dialer and a neighbouring lysine of an adjacent equivalent tetrapeptide or pentapeptide. So this terminal dialer is lost in mature peptidoglycan that's properly cross-linked. The enzyme is called transpeptidase that catalyzes that. And vancomycin, because it binds to the dialer, it's a steric hindrance. It's a large molecule, vancomycin, 1,500 Daltons. There's a stearic hindrance and prevents transpeptidase accessing the dialer and forming the cross-link. So just like penicillin, you get a weakened peptidoglycan because transpeptidation can't occur. And when transpeptidation can't occur, you get weakened peptidoglycan, and as the cell expands and grows, that layer of peptidoglycan, the weaknesses show up a lot more, and it progressively becomes thinner and thinner as the cell grows and grows, and eventually you get a popping of the cell, an osmotic lysis, because the peptidoglycan can't maintain the cell shape anymore. So it's a bit similar to peptidoglycan in the site that it inhibits. It's just acting as a different component of that transpeptidation process in the peptidoglycan. Okay. Other things to note, this is the structure of vancomycin, and you will see that it's made up of some very unusual amino acids. There's only asparagine, which is what we consider a normal amino acid in the structure. All the others are quite unusual, and there are two sugars attached, vancosamine and glucose, to vancomycin. Okay, let's get on to the production. Sorry, I'm just trying to get rid of one of the labels that keeps flying up to tell me I'm presenting. Okay, so how is vancomycin and these other glycopeptides actually produced commercially? Well, they are produced from their producer microorganisms, just as many other antibiotics are, so I've listed some. At the bottom, we've got good old penicillin, discovered by Alexander Fleming in 1928, when he found isolated Penicillium notatum. Now, since then, the industrial strains that are used to produce penicillin have been shifted towards chrysogenum, which is much better at yielding penicillin than the original Penicillium notatum. But similar to penicillin, we have an organism which produces vancomycin, another that produces teicoplanin, and so on. And these are organisms that belong to the actinomycete group of microorganisms. They tend to be very GC rich, so they're a little bit more difficult to manipulate genetically to break the GC bonding that occurs, but it can be done, of course. So this is a process of fermentation of the producer organisms. Yeah, I think that's all I wanted to say. So in these producer organisms, the antibiotics are produced as secondary metabolites, And that's very good news for fermentation producers in terms of yield, because had they been primary metabolites, it would only ever be possible to produce as much product during a fermentation as is permitted by cell growth. So alcohol, for example, is a primary metabolite of yeast. And if you look at the batch growth of yeast, alcohol is produced as part of energy metabolism. So you're only ever going to get as much alcohol produced as there is growth. It's directly dependent on growth. So you see alcohol production following very closely the growth curve for the cells in batch culture. Secondary metabolites, by contrast, are produced not during early and exponential phase of cell growth. They only start to be produced as the cells are starting to level off in terms of their growth following exponential phase. So just as they're tipping into stationary phase, I suppose, near the end of their growth phase, you start to see secondary metabolite production, in this case penicillin. But its disconnection from the growth curve means that you can get really high levels of overproduction in stationary phase. A dramatic overproduction is possible if you manipulate the conditions well enough. You can boost the yield well beyond what the cell biomass is doing. And that's good news. So let's talk about the fermentation process, the industrial fermentations. Fermentation, we think of an anaerobic process, don't we? You know, yeast to alcohol and things. But most industrial fermentations are actually aerobic. So just scotch that myth straight away. And these actinomycetes are aerobic organisms. So it makes sense to have good aeration for growing the organisms. You can do it in lab scale, you can do it to large scale, of course, and we're talking large scale here. But the principles are the same. You have a closed system, a closed vessel. We're talking about batch growth, not a continuous culture where you dribble in nutrients and take the waste products out on a continual basis. This is batch culture because we need that stationary phase region where they are producing antibiotics at their greatest levels. Antibiotics, when we're talking about the size of the fermentor, by the way, we mean the vessel when we say fermentor, but you'd be amazed. Most companies don't use the term fermenter for their vessels. Everything, the organisms and the vessels, tend to be referred to as fermenters with an E. Anyway, anywhere from 1,000 to 500,000 litres is considered a large scale, I suppose. And antibiotics come on the scale in the sort of mid position, anywhere between 40,000 litres and 150,000 litres for antibiotic production. And as mentioned, we have a closed system, stainless steel cylinder, a cooling jacket to remove the heat, or to when you've finished, you want to steam sterilize the vessel, so the cooling jacket helps there too. I've mentioned about oxygen needed, so you have a sparger at the bottom of the vessel, which is a metal plate with holes in it, and oxygen, sterile oxygen, is piped through and enters through those holes. And sometimes that's enough, but sometimes you need help to distribute that oxygen throughout the large vessel. So you have things like propellers or impellers which rotate and distribute that oxygen evenly. And it has the added advantage of distributing the cells really well, and they all get exposed to the same conditions because they're being evenly mixed throughout the culture. Okay. So just to briefly mention what happens once that fermentation process has proceeded for the optimum time in the optimum medium. I'll come back to that in a moment, but I just want to talk briefly about purification before returning to the process of conditions for optimum production. And that is what you'll find is that once the fermenter run is finished, there's initially a filtration step, and you're filtering out the organisms that are producing it, produces a solid, and that is collected, dried, and used as an animal feed supplement very often. The liquid that is left, which is containing your antibiotic, is then captured by absorption in some way on some sort of column. Ion exchange or other adsorption of the antibiotic. If the antibiotic is dissolvable in a solvent, often they will go straight for a solvent extraction of the antibiotic. Then once those processes have happened, you've got a partially purified antibiotic, which is then crystallized and then repurified from that recrystallization solution. And I'll say more about that for vancomycin now. So in the case of vancomycin, these rules all apply. There's filtration, there's adsorption to a cation exchange resin. So that's a negatively charged resin to attract vancomycin, which is positively charged overall at physiological pH. The vancomycin is eluted then by something which is frequently used in industrial physical biochemistry, and that is reverse phase column chromatography. So what you do is you add a more polar, aqueous, mobile phase to the column. And this means that vancomycin, which is hydrophilic overall, is released from the column, the stationary phase, which the column is now preferring to bind more hydrophobic molecules. So the vancomycin is released. Then the vancomycin is crystallized in an acetone solvent, acidified, and precipitated again with organic solvents such as acetone or alcohol to produce vancomycin hydrochloride. Okay, vancomycin has been around for a long time and was discovered during the golden age of antibiotic discovery, which is covered in the previous lecture, the video you watched this morning. So it was discovered a long time ago, 1953, and it was done through natural product screening. So soil microorganisms were systematically collected. This is screened basically for their antibiotic producing capabilities. This is a technique which has very much regained ground. So structure-based drug design was tried. It's still being used, but hasn't been successful so far in discovering new antibiotics. So people are returning to natural product screening, looking what is out there in the environment. Streptomycetes are famous for their antibiotic production, so they tend to be the organisms that people isolate to have a look at. They purify them, they test to see whether they have the ability to inhibit MRSA or other infection agents, and then it's a case of recovering and purifying that biologically active agent, and that's the tricky bit, really. Anyway, this one was isolated from Borneo jungles in Indonesia. I'm terrible at pronouncing this, but I understand it's pronounced Amycalotopsis orientalis. And in soil actinomycetes, vancomycin occur at quite a high frequency. So 1 in every 150,000 that are screened will be a vancomycin producer. Alternative ways, of course, are to isolate the DNA from soil microbes, clone them into cosmid libraries, which are basically plasmids that can take huge chunks of DNA. Plasmids have limits. And you will find that in cosmid libraries, the frequency will be of finding a vancomycin producer will be 1 in 2 million of E. coli clones, which isn't much when you think about E. coli numbers per mil that you can plate out. So you can easily find them. Okay, vancomycin was approved and licensed in 1958, but it didn't get used immediately because at that time there were some things running against it. And so methicillin, first of all, came along at roughly the same time. So to treat penicillin-resistant streps and staphs, people turned to methicillin, first of all, rather than vancomycin as an alternative to penicillin. That was the first event that happened. And one of the reasons they turned more towards methicillin rather than vancomycin, another reason, is because vancomycin has some side effects. So it's responsible for kidney failure. It's responsible for hearing loss in some patients. Now, whether it's the vancomycin or the impurities that were first present in 1958 in preps of vancomycin is still a bit of a matter of debate, but GPs were put off using vancomycin because of the side effects, the toxicity of the antibiotic. It came out a sort of purpley colour, and this is due to, it was called, what's it called, vancomycin mud, something like that. So then it underwent further purification to get rid of those impurities. And some people say that's the problem of toxicity cured by having a purer prep, and other people say, well, it's the vancomycin itself. So it was very underused, but it was the resistance to methicillin which prompted use of vancomycin again in the 1970s and 1980s. So that slide I showed you earlier about the rapid consumption during the 70s and 80s was due to methicillin resistance, and it was also due to the increased prevalence of the C. diff. So oral vancomycin prescription as well has revived vancomycin as an antibiotic, which is used much more frequently these days. Okay, so what about optimizing its production in these fermenters? Well, there's several ways of going about it. And I say at the outset that whether it's by genetic engineering or strain selection or optimizing wild type strains and by manipulating the medium of the fermenter, all these things can be done to optimize yields. So using them all together hasn't been completed yet. So there is still a research area there to take an optimized wild type, generate a strain which can spontaneously produce more. You can still use these optimized conditions and use genetic engineering, and that opportunity is still there to do that. Okay, so let's talk about each in turn. So first of all, we can optimize production in using wild type strains. And I'm not expecting you, by the way, to read up on all these papers. We only have one lecture on this. And there is an excellent reference which covers many of these papers, which would suffice to consult. That's Marconi in 2018, a really excellent review on vancomycin production industrially. So anyway, McIntyre in 1996 produced a medium for the fermenter, which has a strong nitrogen source, high nitrogen source. So peptone as a nitrogen source with added amino acids, and over doubled the yield of vancomycin in flask cultivation, not in his fermenters, but in flask cultivation. So as is very often the case, there will be small scale experiments to see what can be used to increase yields, and then you hope to roll that out in a fermenter. And rollout isn't always successful when you scale up. That is a very important thing to bear in mind. So experimental successes doesn't necessarily translate to a fermenter, but you've got to start somewhere. So Padma et al. Found that he used very defined conditions for vancomycin production in his specific strain. Everyone uses different strains. That's the other thing to bear in mind. So pH 7.6, a specific temperature, a specific inoculum size, very important and underestimated as to what happens with secondary metabolite production. But inoculum size, believe it or not, can have quite a dramatic effect. How much you agitate the aeration conditions, super refined conditions for this particular strain of wild type A. orientalis. Glucose can influence how much vancomycin you get. There is such a thing as catabolite repression, but not in this case. There is an optimum glucose concentration, which is 15 grams per litre, which increased yields. And there was one study which found fantastic yields, 1.6 grams for every litre of culture of vancomycin being produced if you were to trap the cells on a physical support like cotton fibres and glass wool. Amazing yields, though the authors didn't specify what the recipe was for this rich medium, so it couldn't be repeated, but these are fantastic yields. And what I want you to draw from this slide is that using wild type strains, you can get 1.5 grams per litre of medium max from manipulating the conditions that the producer cells are growing in to up your yield of vancomycin. So then we turn to strain improvement, and this scheme just summarizes what that entails. So can't see my pointer. So you can use chemical or physical agents to generate spontaneous mutants that have a higher capacity to produce vancomycin or other glycopeptides. So you can use classical things like UV irradiation. You can use chemical mutagenesis using a very potent chemical mutagen called methyl nitroguanosine, nitrotetraguanosine. I've forgotten what that is now, but it's commonly used as a chemical mutagen. I'll come back to the Protoplast Fusion in a moment. So by chemical mutagenesis, given that the mutagenesis rate is about 1 in 10 to the 8 or 1 in 10 to the 9 cells will mutate beneficially to produce more glycopeptides, then because it's such a small number that mutate in the way that you want, you have to try and screen for them. So how do you screen for something which is able to produce more vancomycin? Well, one of the tricks with glycopeptides is that you have to use associated markers which are associated with increased glycopeptide production. And in this case, it's resistance. So what has been noted is that when you have higher glycopeptide producing strains, this is accompanied by an increased resistance to that antibiotic, to those glycopeptides. Because after all, the producer organisms mustn't kill themselves by suicide through glycopeptide action. They have to have a resistance themselves to their own antibiotic they're producing. And this duly happens. And what you can screen for is an increased resistance to that antibiotic. And the chances are that it will also be a strain that produces more of the antibiotic. So you screen through using clue type phenotypes in the pre-screen. You purify them and check them out that they are actually producing more glycopeptide. And then you can either go straight for industrial scale up, or you can put it through subsequent rounds of chemical mutagenesis and UV mutagenesis, again, to improve it still further. Or another trick that can be done is that you can take these better producers and then just shuffle the genes a little bit, and you might get even higher production. And the way that is done is through Protoplast Fusion. So you take your high producer, you generate protoplasts which are able to exchange genomic DNA, and they recombine it when that happens. And it rejigs, reshuffles, and you might get even better production through that protocol. So that is classic strain improvement. So who's done it and what kind of yields did they get? Well, everyone uses different strains, as I've mentioned. So some of the increased yields might not look that great, but when you look at what the wild type was producing, you can see that you get increased yields. So Young et al. Were the first people to do this using this strain, ATCC 19795, which is used commercially today. And they did UV mutagenesis to create a new strain, which, when they grew it on this concoction of substrates and under these conditions of oxygen, etc., they got yields which were tenfold higher than the wild type, which is pretty impressive. And they were then isolating vancomycin at a yield of 11.5 grams per litre of culture. And you can imagine how much you get when you scale that up to much larger industrial scale. And Zheng et al. Used a UV mutagenesis protocol 2 with a different strain. And this is where in the recent literature you see people are starting to use sustainable feedstocks to grow their producer organisms in fermenters. So now we've got biodiesel derived glycerol, potato, protein, thinking about sustainable feedstocks and doing their studies in them to see what kind of yields they get. They got an impressive 7.6 grams per litre in their shake cultures. And as I've mentioned, when you scale it up to fermenter, you don't always get success. So it's still good, but it's not as good as in shake culture. And that's such a common thing you see in these sorts of scale-up experiments. Nonetheless, it's good. Okay, there are other things you can do to trick the producer cells into producing more glycopeptides. And one of them is called ribosome engineering, which Tanaka et al. Used in 2013. So you can select for spontaneous mutants that are resistant to rifampicin. Okay, rifampicin is an antibiotic which binds, it acts on the RNA polymerase of microbes, specifically binding to the beta subunit of RNA polymerase. And when it does that, it shifts the specificity of the polymerase for recognizing certain genes. So it switches the specificity to transcription of secondary metabolite genes. And what it's doing is mimicking what happens in vivo. So rather than rifampicin, what happens in vivo is that these organisms are able to shift transcription to secondary metabolite genes through something else, not rifampicin, but something called PPG-PP. When that binds, just like rifampicin, when it binds to RNA polymerase, it favors secondary metabolite gene transcription in vivo. What is PPG-PP? This is a collective term for guanosine tetra and pentaphosphates, which occur in the cell, and in times of stress, these are produced and they bind directly to RNA polymerase. Beta subunit is heavily involved in that binding, and this regulates the promoter selection for these secondary metabolite genes. It also stabilizes the sigma subunit S, which is the specific sigma subunit of RNA polymerase required for secondary metabolite genes. So there's a twofold effect, and rifampicin is a way of artificially creating the same thing, the same effect. Okay, so Tanaka et al. Did that. They selected their rifampicin resistant strains and found that they could boost vancomycin production as a secondary metabolite 2 to 3-fold over the wild type. So these yields don't look as good as what we've seen previously, but it is nonetheless a 2 to 3-fold boost in this particular strain that they used for their studies. So again, that this has been a successful strategy. I'm going to return to this later as well in the genetic engineering section, which is coming up now. Okay, so you can use rational genetic engineering for strain improvement too. How are we doing for time? Not too bad. Okay, so let's take each of these in turn. Okay, so scenario number 1. As prokaryotes, these producer strains tend to group their glycopeptide production genes together in a big cluster on the genome. So related functions tend to be clustered, and glycopeptide production is no exception. The genes are present together on the chromosome, and they are subject to positive regulation at the transcriptional level. So they have one promoter region which is subject to positive regulation by regulators which bind to the promoter and activate transcription of those glycopeptide production genes. So we're capitalizing here on a prokaryotic feature to overexpress these activating transcription factors. They are typical activators of the STRR and LuxR group, and I've just made a note of their characteristics below in case you want to look them up, but they are classic activators. In terms of teicoplanin production, they are encoded by Ti15 or Ti16, these activator genes, these positive regulatory genes are cloned, and they are cloned into a plasmid which integrates within the producer strain genome, far safer than letting the plasmid replicate separately from the genome, which tends to be an unstable event in large-scale fermenters. So you integrate the plasmid carrying these activator genes, and let them integrate into the genome. If those genes are under a constitutive promoter, such as the AAC promoter, it means it's switched on all the time. So production of Ti-15 or Ti-16 is happening all the time. So there's more activator being produced, and therefore more transcription and translation of the downstream glycopeptide production genes. And when this strategy is used, then you get a 10-fold boost in production of teicoplanin as a result of that. So you can get well over a gram per litre using this strategy. So that's number one. So number two is it's what I've covered before. It's this idea that when you get increased production, you get increased resistance of the producer organism to the antibiotic. So if you increase the organism's resistance to the antibiotic, this is accompanied by higher yield. So it's as if you have to overcome the barrier of the producer organism, the resistance, in order to get higher yields. It's like a barrier that has to be overcome, and then you can increase your yield if you increase the resistance of the strain to the antibiotic. So what is the resistance to the antibiotic in the producer strain? Well, one of them is Van-Y. Van-Y is part of the resistance operon to glycopeptides. And Van-Y is a carboxypeptidase which cleaves off the terminal D-ALA in the Pepto-Glycan. So if Van-Y is produced, that peptidase then acts on the Pepto-Glycan, cleaves off the terminal D-ALA, which means that vancomycin can no longer bind to that area because it needs a D-ALA to bind, not just a single D-ALA. So effectively, that Pepto-Glycan has become resistant to the action of the glycopeptide. So if you overexpress the Van-Y, the hope is that you will then, by increasing the dosage of Van-Y, you will then increase the resistance, and that this will be accompanied by the higher yields, however that happens. And this has found to be the case. So the study so far, first of all, knocked out the chromosomal indigenous copy of Van-Y, that the organism does survive. It's less resistant, but it's okay. And then they added plasmid-borne copies, in other words, multiple copies, to vary the gene dosage. So by doing that, they increased production by up to 2-fold, or even 3.5-fold over the wild type there by that mechanism. So it's an accompanying thing. If you can increase the organism's resistance, you can increase the yield of glycopeptide that the organism will produce. Okay, mechanism number 3, or strategy, genetic engineering strategy number 3, is by overexpressing a chaperone, which is involved in producing the glycopeptides. So we have to know, first of all, that glycopeptides are synthesized by non-ribosomal peptide synthetases known as NRPSs. But in order for these NRPSs to produce glycopeptide, their substrate is aminoacyl AMPs. rather than amino acids themselves. So they need amino acyl AMPs in order to produce the glycopeptides, that is the substrate. And in turn, these amino acyl AMPs are produced from amino acids by adenylating enzymes, of course, which are going to use ATP to attach the AMP moiety onto these, or the acyl AMP moiety onto the amino acid group. So adenylating enzymes work if they have a chaperone to fold them correctly. So here are the adenylating enzymes, and they require to be folded properly in order to function, to become active. And it is this MBTH chaperone which does that job and folds the proteins correctly so that the enzymes work, produce the substrate, which in turn produces the glycopeptide. So what researchers have done is to overexpress this chaperone. They've put it under an promoter, which is, I think, constitutive, and put it all into an integrative plasmid, which will insert the MBT gene under a strong promoter on the genome, significantly increase the chaperone and therefore the amino acyl AMP pool. And indeed, this did result in an increased production of vancomycin. And they did this in an already high producer. And this is what I mean about piling on different strategies, one on top of the other, to increase yield. So this was already a high producing strain of 3 grams per litre, and they managed to increase the yields just by overexpressing the chaperone to 5.3 grams per litre. So that has been another strategy. The next strategy, number 4, is to overexpress S-adenosyl methionine synthetase, SAMS. And SAMS is known to stimulate secondary metabolite production in Streptomycetes in general. Okay, so what does this synthetase do? Well, it takes methionine and ATP to produce S-adenosyl methionine. And the significance of that is that S-adenosyl methionine is used as a source of methyl groups to be added on to secondary metabolites during their biosynthesis. So if you can overexpress the gene for SAMS, which is MET-K, and again, put it on an integrative plasmid, constitutive strong promoter, put it into the teicoplanin producer, actinoplanase, which I can pronounce better, then when you do that, again, you can increase teicoplanin production, in this case by 2.2-fold. We're almost there now. The last 2 strategies of genetic engineering occur without exploiting the biosynthetic gene clustering, and that is to overexpress or change the sigma factor of RNA polymerase and change it by error-prone PCR. So the sigma subunit of RNA polymerase is the subunit that recognizes the DNA recognition sequence to initiate transcription. So by selecting for changes in the encoding gene for that sigma subunit, you can improve the efficiency of that sigma subunit to produce higher levels of teicoplanin. And again, that has proved successful in increasing yields to about 5 grams per litre teicoplanin. So changes in the we've covered earlier about the beta subunit of RNA polymerase earlier on. So you can use changes in the RPOB, the beta subunit of RNA polymerase, by rifampicin resistance selection as well. Earlier on, we talked about that. So using the two together could also be another strategy for the future. So error-prone PCR to change this sigma subunit together with the beta subunit of RNA polymerase is another strategy. And finally, you can tailor the enzymes that decorate the vancomycin scaffold to generate new varieties and variations in the final glycopeptide that's produced. So gene knockouts have also been used to alter the glycosylation patterns of the final glycopeptide that's produced. So I've given you 3 examples here. TIE 10, 11, and 13 encode these enzymes which add on these groupings onto the glycopeptide structure. So you can delete them out and get different combinations of glycosylation patterns on the final antibiotic, and that's ongoing kind of strategy to develop novel glycopeptides. Okay, so I've run through what I was going to say today. This is just acknowledgements and the main reading. So there's some good books which cover the sort of fermentation side of things, the clinical use of antibiotics, the information that I've used, the background information that I used for the early parts about antibiotic usage, and this lovely review by Marconi that I mentioned earlier that goes into the industrial production. So everything under one roof here in terms of reading up about the genetic engineering, the classic strain improvement, the optimization of wild types is all in that one reference. And if you want to learn more about the history of vancomycin, which has had a very up and down history in terms of its usage, Levine is very good if you want to read some of the background there. So that is it. That is all I was going to say. So if you have any questions,